Hello, my name is Eus Van Someren, and I'm very happy, very glad to, with the invitation to tell something about what my team has been looking for. We've been in search of brain mechanisms of insomnia. It's a joint effort with many people. I don't have anything to declare, and if you are running out of interest or time or concentration, I made the first slide such that you have the gist of the talk already. So what I'll try to demonstrate is how my search for brain mechanisms of insomnia suggests a hypothesis that there is something going wrong with memory trace plasticity in REM sleep. And what's going wrong entails overnight disturbance of stress and emotion regulation. So normally you see this baby on the left. A lot is going on in REM sleep. There's reactivation of brain circuits, as you might see. And these include especially emotional brain circuits or limbic circuits. And they do a lot of good. But the question is whether that works for everyone, and especially for people with insomnia. So this is an overview of what I will be talking about. First, I will be addressing the question why to study insomnia at all. Then I'll take you by the hand in our search for clues on brain mechanisms of insomnia. I'll explain a little bit on how we stumbled upon heterogeneity and subtypes of insomnia. Then how we developed the hypothesis of failing overnight distress adaptation. Then an example from another field. We could learn from how phobia research and treatment has been developing and gave some clues on how to get rid of a stuck bad memory. Subsequently, I'll move to restless REM sleep, to what extent that could be involved in the risk for emotional problems. I'll give you a cartoon of the model that we are developing and testing. And finally, if there is sufficient time, I will address a little bit on whether we can ameliorate the risk for emotion disorders if we treat insomnia. So why study insomnia? The question that I like to address. What you see here in this cover of Nature Human Behavior early this year, there was a large study from my lab together with many other labs indicating that poor sleep quality and insomnia symptoms are much more prevalent than short sleep. And this was somewhat surprising because most of the guidelines in the general media, they stress that we should address optimal sleep duration to prevent short sleep. But our findings highlight the importance of targeting sleep quality instead. Moreover, sleep quality is the primary modifiable factor determining the risk, severity, and relapse of the most prevalent mental disorders, including depression, anxiety, and PTSD. And you see here this graph indicating the prevalence of the different categories of disorders. We see the combined anxiety disorders on number one, then insomnia, and only then depression. It's often, insomnia is often left out of these kind of overviews, and that's a pity because we think, and I will try to convince you of that in my talk, that insomnia could be pivotal to these other disorders. So in the Netherlands, this observation has led to a knowledge agenda that we should prevent and treat insomnia and the risk it conveys. But to do that properly, I think we need clues on brain mechanisms to improve treatment. So if we want to have clues on brain mechanisms, let's first start on insomnia vulnerability. So how does this develop? Well, as you know, probably genes and early life stress do a lot, and that's also the case for insomnia. To start with genes, we know from meta-analysis that heritability is about 40 to 44%. So part of the risk, whether or not you will develop insomnia, is already there at conception. In collaboration with the team of Daniela Postema, we did genome-wide association studies, and we found a strong correlation of genetic risk of insomnia, especially with anxiety and depression. So genes that put you at risk of developing insomnia overlap to quite some extent with genes putting you at risk of developing anxiety, depression. So that raises the questions, what is common about anxiety and depression and what is different? So we all have our occasional bad day, but what differs in these disorders is that those bad days, they do not resolve overnight. Emotional distress is just taken to the next day, night after night. So sleep brings no relief. And the clue we have here is that there may be some failing overnight adaptation to emotional distress. May sound trivial now, but I will get back to that. And that's why I put it in red font here. I will do so with all the clues we can get. We can get more clues from GWAS. So one thing we can do is, if we found a lot of genes that are associated with insomnia, where variants can put you at higher risk, then we can subsequently see, by using other databases like GTEx and a database in the Karolinska Institute, where these genes are actually active, in what tissues are they expressed, and in what cell types are they expressed. This is just to explain how that could work. Suppose that these are a number of genes that are expressed in insomnia or change the risk of insomnia, and you find a particular cell X that doesn't express any of these genes ever in its life, then it's very unlikely that this cell X is mediating the genetic risk of insomnia. In contrast, cell Y expresses several of these genes where variants put you at risk of insomnia. So it would be more likely that cell Y is somehow involved in the genetic risk of insomnia. So this is how this works simply. So this has been done by Philip Janssen, published in Nature Genetics in 2019. And what we found was several areas, cortical and subcortical, where we could pinpoint gene sets that are put you at risk of insomnia and are expressed in these tissues or cell types. And there were a number of things that were really remarkable. So first thing is that we found something in the prefrontal cortex. And that was the first site ever found to be involved in insomnia by Altena a long time ago, 2008, I think. But an interesting other thing was that there was elevated gene expression or expression of where variants put you at risk of insomnia in the anterior cingulate cortex and also in the claustrum. And I was excited to read this because work of the lab of Hervé Lupi demonstrated that specifically during REM sleep, these neurons in the claustrum over here activate the anterior cingulate cortex. So these were two areas that caught our interest, especially because they are so linked during REM sleep. And I will get back to this later in my presentation. So moving from genes to early life stress, there is prenatal stress, of course, and the bottom line is that we're not very sure to what extent early life stress, prenatal early life stress, increases the risk of insomnia. There are some observations, but I think the bottom line is we're not very sure about this. On the other hand, childhood adversity has lifetime effects on sleep. What we see first looking at sleep is that there are more arousals. And again, pointing a little bit towards REM sleep, this is most remarkable so for arousals in REM sleep. If you look at specific insomnia diagnosis, we see that the risk increases most by frequent fear and serious conflicts, repeated and diverse adversities, and lacking simultaneously safe parental bonding. And this again generates a clue for us. So first, of course, these REM sleep arousals that drew our attention, but also we have the idea that learning not to be safe during a critical plasticity period may strengthen brain circuits that support watchfulness. So if you're just trained to have to be watchful all the time, or many times if you're a kid, maybe you develop circuits, brain circuits, biased to being watchful, staying watchful, which comes in handy if you're in a dangerous environment, but may not come in handy if you want to sleep, especially later at life when sleep becomes more vulnerable. So could we find such brain circuits then involved in watchfulness and not insomnia? Well, if we move to imaging findings of insomnia, it was sort of, we were not too happy to find that there was very inconsistent results. Several labs found other things, and I gave them here several colors. So, for example, increased thalamic activity and reduced caudate nucleus recruitment, alterations in anterior cingulate cortex. Again, we've seen it again before. Amygdala, hippocampus. Well, this is not, it's not really nice if different labs find different things, but that made me think of a finding in another disorder. And in that disorder, they said, well, there is not one type of, I think this is ADHD. There's not one type of ADHD, but there's clusters of patients that resemble each other and that are a bit further away from clusters of other also patients that do resemble each other. So it might be, it was our hypothesis, that maybe some of these brain areas are involved in one subtype of insomnia, while other clusters of patients may have problems with another part of the brain. So this is a hypothesis we wanted to evaluate. And at the time we started that, about 10 years ago, there was no nice database of brains that we could evaluate on risk of insomnia. So we thought, well, let's start then with something that is close to the brain, but not really the brain. So this is the hypothesis that there are several profiles with genetic makeup. So there could be several ways that could affect the proteins and microstructure and macrostructure of the brain and brain function. And ultimately, you could have a complaint. And the complaint may be the same for all these subtypes, but the underlying mechanisms may differ. So that's the idea. We wanted to start with brain data. They were not available at that time. And then we thought, well, we know of some of these brain structures that they are related to stable traits. For example, neuroticism or perfectionism. So what we did is said, okay, let's try to find a lot of data in a lot of people about biologically based traits that are relevant to insomnia. We did that with a website called www.sleepregistry.org. And the idea was not to get an average blurry profile of the average insomniac. No, in contrast, what we wanted to do is disentangle that blurry overall average and see if there are different subtypes of insomnia there. It can be done in a data-driven way with latent class cluster analysis. So we call them multivariate subtype fingerprints. Then not just in the brain, but in traits that are related to brain differences. And then if you would have them, of course, we could go to the other more expensive measures like high-density EEG or recording in the MRI scanner or TMS or whatever. The bottom line is that we found 5 subtypes, and they are shown here, and they are not defined by one single variable. They are defined by a fingerprint of variables. So you see here along these spokes the different variables like negative affect, lack of subjective happiness, rumination, neuroticism, etc. And in the colors you see the profiles of the different subtypes we found. So they have specific fingerprints. This classification was robust. That meant there was in general no doubt. The subject wasn't half subtype 1 and half subtype 3. If we could find a subtype, it was usually with a high probability near 1. People also didn't change. We followed them up over several years, and we found that in general they stayed in the same subtype. And it was generalizable. We did it in a new sample, and we again found that same 5 subtypes. It's highly relevant, we think, because these subtypes differed specifically, for example, on lifetime depression risk. I'll get back to that. That differed even up to a factor 5. Some people with insomnia have no elevated risk of depression. Others have a risk of lifetime depression that's even more than 0.5. What was remarkable, we had measures about clock and homeostat, questionnaire about it, but these subtypes were not defined by these clock-related or homeostat-related variables. The clues here that we get from the subtyping is that they point to affect, to reactivity, and to reward sensitivity traits. So that's maybe more where insomnia is present. And for 2 of these subtypes, we just happened to have some EEG data. These were what we called, based on questionnaires, moderately distressed insomnia. And the other one was low distressed insomnia. And what we had available was an oddball task. And I don't know if you're familiar with an oddball task. It's you just play sounds, very boring sounds. I will give you an example. So that's really boring, right? And you see that mostly there is this low sound, and every once in a while there's a deviating sound. And if you make an average brain response using EEG, you see that this is the response to the regular tone, and this is the response to the deviating tone. This is a sort of aha of the brain. There is something that I should pay attention to because it's not regular. So this is good sleepers, and then we go to one subtype of insomnia in green here, and you see that there's really nothing wrong with these evoked potentials in this subtype. But if we move to another subtype, the subtype that we called reactive type based on questionnaires, we see that adaptation fails. So even with the regular tones, you see that there is this deflection here, about 400 milliseconds, indicating that people label this stimulus as salient, as important, as requiring attention. And what's maybe even worse, if we look at this late potential here, also in purple, that was also significantly different, is labeled as emotionally relevant. Just imagine that something trivial as these tones are labeled as emotionally relevant. That's this late positive potential. Well, that's remarkable. We have this clue to failing adaptation and labeling regular input as salient or emotionally relevant. But what's even more worrisome is that this occurs even for every own heartbeat. We did the same type of research and generated an average evoked potential triggered by your own heartbeat. So we don't provide any stimulus at all. And we see here late in the average response that there is this difference between insomniacs and controls. As if an insomniac pays attention to each heartbeat with a sort of idea, aha, I should pay attention. I should pay attention. I should pay attention. This is something that can be very tiring, as you can imagine. And we also found that this involved medial prefrontal cortex, so that's an area involved in attention. So these were very simple examples of brain function with evoked potentials, but there's of course also, let's say, higher brain functions. And we know, for example, from sleep deprivation studies that, for example, or in particular, attention is very sensitive to sleep deprivation. You see here a meta-analysis, that is, this is the average decrease in alertness, as you can find, relative to 0, relative to having had good sleep. And if you look at all the individual studies, there is just no doubt that sleep deprivation does something to this cognitive skill of maintaining alertness. Now, if you move to a similar meta-analysis for insomnia, again a forest plot, now it's different cognitive function. And as you see of these individual studies, they usually include the 0 line, indicating that people with insomnia do not differ from controls. This is actually, I think, remarkable. In spite of their often years-long complaints and poor sleep night after night, they perform really relatively well. The one thing that was most reliably affected insomnia is episodic memory. So clues that we have here is that, well, insomnia is really something different than sleep deprivation. There is certainly no lack of alertness in insomnia. And what's most affected seems to be especially self-referential memory, because it concerned episodic memory, not, for example, procedural memory. From this, we have some more clues, indirect, of course, because if you think of what memory is adapting synapses, like visualized here, in neuronal networks, like visualized here. And this is something I will get into later. I almost forgot that maybe from sleep we could also have some clues about what's wrong in insomnia. Polysomnography is not usually done in insomnia, but if you do so, this is data from Dieter Riemann's group, Freiburg. This is a well-sleeping subject. It's visualized in colors, wakefulness, REM sleep, stage 1, 2, 3. And then if you look at the person with insomnia, there's certainly sleep, as illustrated by red, green, blue. And what's most distinguishing for insomnia relative to control is that, the pattern of a lot of interruptions of sleep. So arousal is actually going on during the sleep period. And it has been found that it's even most marked in REM sleep. So again, a clue towards REM sleep and ongoing arousal during sleep. So to give a summary of the clues we have so far and to try to frame a hypothesis, well, we found that insomnia might be related to failing overnight dissolving of emotional distress. It might be that during development there is a particular well-developed brain circuit that support watchfulness. So if you have to be watchful all the time in that time of plasticity in childhood, then maybe you become very good at that, better than you would like to. We didn't find too much going on in the clock in the homeostat, so instead of sleep regulation, we might think of other things like affect and reactivity. And we had some indirect clues that maybe there is something going on with synaptic plasticity circuits, and maybe especially those circuits that are activated in REM sleep. So this generated a hypothesis, and the hypothesis is that there might be failing overnight adaptation of emotional distress and memory. That because of that, people take their distress to the next day, every day, and they become sort of stuck in being aroused, hyper-aroused all the time and not being able to get rid of that. So I'll give you a little bit of background on how we got to that hypothesis, that failing overnight adaptation hypothesis, because that was in part inspired by people that investigate stuck dysfunctional anxiety associations in anxiety disorder. So I'll take a detour. So I deviate now a little bit from insomnia, and I'll tell you what can go wrong if there is a stuck dysfunctional association, fearful association. Well, let's start here. If a dog like this is approaching you, then it would be really handy that you will be fearful and you stop doing whatever you were doing and just make sure that you're safe. So just seeing and hearing this dog immediately without any thinking activates the locus coeruleus. Noradrenaline is released throughout the brain. This is, by the way, mouse brain. And what happens, whatever brain circuit was active, limbic and salient circuits will take over. They are prioritized and they make connections everywhere. So that's what noradrenaline does. For example, if you're experiencing fear. Now, there are people that have what we call a dysfunctional association. They have developed phobia. For example, if you see this nice fluffy thing here, and then you realize that it's a dog, then immediately you become extremely fearful again. Well, there's no reason for that at all. Now, what happens if you become fearful again is that synapse that connects this visual and auditive impression with your fear circuits is labile for a moment. It's amenable to changes. Now, there is this trick that has been applied by people trying to help people that have these dysfunctional associations, and that is to give propranolol after exposure. And it's thought that what propranolol does is, so it blocks any effect of noradrenaline, and it can break that association. Some remarkable successes have been there by just doing this treatment, and the synapse that mediates this dysfunctional association is depotentiated. So that made me think of something. So noradrenaline timeouts. In people with working with anxiety patients, this is done by propranolol. But now let's go back a long time to study Gary Aston Jones, published 40 years ago. This is multiple unit activity, so activity of cells in the locus coeruleus of rats, meanwhile recording EEG and recording EMG. It's visible here if the amplitude of the EEG increases, that is slow wave sleep, and you can follow also EMG here. But it doesn't matter if there's wakefulness or slow wave sleep, there is activity of the locus coeruleus. And it's only when the animal enters REM sleep, here, this small arrow, that the animal experiences a long period of no activity in the locus coeruleus. And then there is wakefulness again, and here you see that locus coeruleus activity kicks in again. A recent study by Kierby sort of shows the same, but with more advanced methods and more control over everything and more recordings locally. Here you see locus coeruleus neurons, their activity, and then particularly in REM sleep, that's these greenish areas here. If you measure downstream noradrenaline, the medial prefrontal cortex in this case, you see that there is this slow decrease with the time constants of noradrenaline. Here again, here again. And as soon as there is any interruption from slow wave sleep or wakefulness, you see that immediately that decrease in noradrenaline is halted. And again, the brain is getting higher. So this is a sort of summary of the study of Gary Aston Jones. You see here that activity in the locus coeruleus, only during paradoxical sleep there is no activity. And this is another way of visualizing it. Then for noradrenaline, that's downstream in the cortex, for example, during wakefulness, during slow wave sleep, and there's nothing in REM sleep. But if now we go back to people with insomnia, I've shown you that they have a lot of arousals from REM sleep, so they may never reach these low levels of noradrenaline. And I visualized it here as indicating that even during REM sleep, they may not have a timeout of noradrenaline. So is that important to miss that timeout of noradrenaline? Well, it might well be so, because noradrenaline differs, presence or absence of noradrenaline differs a lot with respect to synaptic plasticity that's going on. So in the presence of noradrenaline, there's a sort of prioritizing of low long-term potentiation, as we see in wakefulness. Now, during REM sleep, which is restful and does not have all these arousals and has a low level of noradrenaline, there may be a better balance between long-term potentiation and depotentiation. And this has been related to fear extinction, so it would be functionally relevant. This is a model by Gina Popa. Now, if you go to, for example, her PTSD rats, which resemble insomniacs in a lot of ways, there is noradrenaline present during REM sleep, so not the way it should be. So there is not this balanced LTP and DP depotentiation going. There may even be fear enhancement, is the hypothesis. So there is a lot of other studies going on that sleep without noradrenaline timeouts might disrupt plasticity and adaptation. And we think if that occurs in REM sleep, it's especially in the REM active circuits. And these are the limbic and salient circuits. Well, the limbic and salient circuits were among the clues that I gave you in the first part of my presentation. So we're getting to a sort of hypothesis that this might be going on. So suppose that we have this circuit of the locus coeruleus and then connected to amygdala and all other kinds of cortical areas like orbitofrontal cortex, insula, anterior cingulate cortex, also seen as relevant in insomnia. In good sleepers, prior to falling asleep, there's noradrenaline. If they have slow wave sleep, it drops a lot. If there's REM sleep, it's virtually absent for a while, and then they return to slow wave sleep and it stays low. But if you think about the type of sleep that insomniacs have, and also the type of wakefulness, noradrenaline might be higher, drop maybe a little bit, drop maybe a little bit more during REM sleep, but not to zero. So maybe this could interfere with processes that go on, synaptic processes that go on in limbic and salient circuits during the night. Another way of visualizing this and framing it is that we think that a sleeping locus coeruleus, so really sleeping, not being aroused all the time, could facilitate overnight emotion regulation in this way. Suppose you have a stressful experience, then you're allowed to have a nap or sleep with consolidated REM sleep that somehow helps you remodel all these synapses that are relevant in these circuits, and you feel fine again after sleep. What we see in people with anxiety disorders, insomnia, depression, and PTSD is restless sleep, and in particular restless REM sleep. And maybe this is something harmful to that overnight plasticity. And it might stay, the distress may stay, or even worsen overnight. So how are we going to study this? Because I've been telling you a lot of modeling work and thinking and evaluating stuff. But how can we address this question? Well, this would be the hypothesis. So if you would stress someone in the MRI scanner prior to sleep, you expect activation of limbic and salient circuits. I show here amygdala. Then if you allow restful sleep, then you can imagine that there would be a decrease in activation if the next day you think about that stressor or are again exposed to that stressor. So here you do not see that clear activation of the amygdala anymore. Now, if you would do that with people with insomnia, that's the hypothesis. We give a stressor in the evening, we measure amygdala activation. They don't have that timeout of noradrenaline during REM sleep. And the next day, if we stress them again or ask them to think about, they again show the activation of limbic circuits as if they didn't have any sleep at all. So then the question is, how to experimentally induce emotional distress? Psychologists among you may say, well, just grab the International Affective Picture System. But I wanted to do that first, but then I asked myself, is this really doing what I want to do with these patients? So this is a picture from that International Affective Picture System. And I wonder if any of you looking at this video feels your heart pound, your hands sweat, your stomach contract. I don't think so. For me, it triggered an emotion, but it was a happy emotion. And that was because I thought of Pulp Fiction when I saw this video. And that was a happy memory. So that was wrong. That was not the intention of the International Affective Picture System. And thinking about that, what's relevant to mental health is not so much these basic emotions, but much more these self-referential emotions. And because you saw, I explained in the first part, that especially self-referential memories, like episodic memories, there seems to be something wrong in people in some way. This is what we wanted to do, to induce self-referential emotion in the MRI scanner. We started piloting in doing so. The question is, how can we do that? So this is the model. Give a stressor, have good sleep, give the stressor again, see what happened. Was there any extinction of the distress? Maybe there wasn't good sleepers. And insomnia, maybe not. That's the hypothesis. But how can we do that? The way we did it was to induce shame, embarrassment. This is a self-referential emotion, self-conscious emotion. And what I will do is I'll let you hear how we did that. So we recorded karaoke singing subjects. They didn't hear themselves sing because they had these big muffles on their ears. They heard other singers and they heard loud music, but not themselves. So they started to sing out of tune and out of beat. And then we recorded their voice without the music and without the other singers. And this is what it sounds like. So I don't know how you take this, but if I hear this, I have some sort of shameful feelings. It's not my own recording. I forbid Rick Wassing, who did these studies, to share my recordings. So singing out of tune and out of beat, and then hearing your own voice, yourself doing that, generates shame and embarrassment in people. And I think it's a reliable way of doing this research. So this is what we found. This is green. This is good subjects playing in the evening and playing in the morning. First asking them the shame it elicited. And we saw that in good sleepers, a night of sleep helped them to think about this was less embarrassing, less shameful. There was this significant drop. Now what we found in people with insomnia, much to our surprise, we thought there will be probably a straight line, no change, no benefits of sleep. But it seemed as if sleep was even harmful, and this happened only from the evening to the morning. It's published in Sleep by Rick Wassing in 2019. So it seems as if people with this fragmented REM sleep and fragmented sleep in general may even enhance or become more sensitive to a distressful experience or thinking about. So from here, of course, we wanted to have a look in the brain. That was the purpose of finding this protocol, and we put people in the scanner prior to and after sleep. We played this shameful stimulus, and we checked overnight whether there were changes in amygdala activation. And there were indeed, depending or associated with the time spent in REM sleep. The more time spent in REM sleep overnight, the more there was a decrease in amygdala activation while hearing this shameful experience. Now, this was the case particularly so for these good sleepers with consolidated REM sleep. But the kicker was that people that had really disturbed REM sleep, like shown here, fragmented REM sleep, the decrease in amygdala activation as shown here in this green area for these people, consolidated REM sleep, turned towards even a small increase in amygdala activation overnight for the people that had the most interrupted, most fragmented, most restless REM sleep. So REM sleep can be helpful, but if it's fragmented, it can be detrimental. This might still be seen as a correlational study, but to overcome that, we did also targeted memory reactivation, which boosted both favorable and adverse effects of REM sleep. So if you had consolidated REM sleep, amygdala deactivated more overnight, and if you had fragmented restless REM sleep, there was even an increasing sensitivity of the amygdala overnight. Now, the second thing we thought about is most of the insomniacs we see, they have insomnia night after night. It's not a single night. They have that often for 10 years or 20 years or 30 years. What would happen with memories of old distressful experiences, like listening to your own voice and being very shameful? So we made that comparison, and here I show another area. This is again the new distress and the karaoke experience, but we also asked good sleepers to go back to distress they experienced 10, 20, 30 years ago. That was also very shameful. They didn't have to share it with us, but they recalled it in the MRI scanner. You see that the activation of anterior cingulate, for example, on that salience network, is not happening anymore. It might be under control of the medial prefrontal cortex and not activate anymore. So that distress, thinking about things that were once distressful, they don't give new distress. But if you look at people with insomnia, there is this activation again, and it's a bit more than in people without insomnia. But also if we ask them to go back 10, 20, 30 years ago to similar distressful experiences and think about that, we see that the limbic and salient circuits activate again, as if it just happened now. So this sort of strengthens our belief that maybe the limbic and salient circuits, which are really active during REM sleep, if there is restless REM sleep, it might interfere with normal processing, normal overnight plasticity. So I'll give a summary of how we think about this in a cartoon. So suppose you experience a very distressful accident. You are in the street, you see something happening that's really awful. Now, of course, you see it happening. You see the car, you hear the sounds that are awful. So it's, let's say that's facts, it's vision, it's auditory information. And that's represented in a network in the brain. I made it blue here. So this is just that network representing, let's say, the cold fact. But meanwhile, if you see this, you have these shaking legs and you feel your heart pound. You might have weak knees, stomach knots, whatever. And these, of course, are also represented in the brain network, in the limbic and salient network. So there is this distress, this bodily distress, and there's this vision and audition. And they're linked because they happen at the same time. Stressful experience link neuronal networks representing facts and arousal. I don't know if you ever experienced that, but if you then think about it 1 hour later or 2 hours later, if you tell someone, many people again feel that distress. And that's not surprising because there is this network of, you could recall that event, what you saw, what you heard, but also that's linked to how you felt. So you might have this memory trace if you recall the fact that arousal, that bodily arousal may be triggered again. Now here comes the nice thing of restful sleep. If you have restful sleep, that helps you replay memory traces and adapt them. And in REM sleep, there is specifically these traces that involve salience network and limbic networks, so that the next day, this is the hypothesis, if you recall the event, you will be able to do so. Still somewhere, stored somewhere in that circuit that was about the facts that you saw, vision and audition. But it will not trigger again that bodily experience. You will not have the weak knees again, the pounding heart and sweaty hands. Now with insomnia vulnerability, there is this idea that, well, maybe what's happening here in REM sleep, there's not this drop of noradrenaline. So whatever weakens these purple connections here between blue and red, they are not adapting in a natural way. And the idea is that if you recall that the next day, that again, this may trigger, the recall may trigger again this bodily experience. So if you think of this is an extreme example, but if this would occur to you day after day, then it would be easy to imagine that with whatever you think about or whatever you imagine, there will be triggering of bodily arousal. This could underlie the general finding that people with insomnia are hyper-aroused all day and night. So if that accumulates, you can also think about what happens then. And I think that what could happen is that if this goes on, it could predispose you after a stressful event to sustained stress and maybe even anxiety, depression. This brings me to the sort of overall model. So there is risk genes that increase your risk. We don't know all of them and also not their interactions, but they're there. And there is also heritability, clearly. There's early life stress. And what we think is that this predisposes you to restless sleep. And in early life, there may not be clinical symptoms yet, but there are some indications, also from my own team, that in early life you can measure already alterations that indicate restful sleep. Not only in my own team, also in other teams have seen that early life stress may lead to restless REM sleep. It's reviewed in a physiological review that's out in July 2021. So if that happens for a long time, there may be insufficient dissolving of emotional distress. And the first thing that might happen then is you become hyper-aroused continuously and be an insomniac. And if that continues, other emotions may get really stuck and you might develop PTSD easier or anxiety disorder or depression. So this is a working model. And then, of course, the question is, can we do something about it? Well, I think we can. And first observation here is cognitive behavior therapy for insomnia. It's, well, the first line treatment for insomnia. And if you think about restless sleep, so most of the restless REM sleep is at the end of the night. So what you could do is, for example, take out part of that last part of the night. This is something that Rick Wassing now in Sydney did. Just sleep restriction, which is the most effective part of cognitive behavior therapy for insomnia, induces about immediately induces a strong decrease in restless REM sleep immediately overnight after one night. So this is one thing we can do. So I want to go into detail a bit more about cognitive behavior therapy because I was about mechanisms and I think we need to do more for insomniacs, and I will explain why, and that closes my presentation. So this is a study from Charles Morin, already more than 10 years ago. If people have insomnia to start with, sleep impairment is about 22 here. We can do nothing, that's the red line. Well, then not really a lot happens. We can give people pills, that helps a bit, but in the end, not that much. Or we can give cognitive behavior therapy for insomnia, which helps a lot, as you see here. You can also give both CBTI and pills, but that isn't any better. So this seems all very nice, but there is a caveat. It doesn't work for about 30% of the people, that's the idea. It alleviates the complaints by 30 to 50%, but in a meta-analysis by Seifert in 2016, it was shown that on average, the sleep efficiency stays below 80%. So it's no way that these people are treated completely satisfactorily. To give you an idea of what this would mean, I made this comparison with fever. Suppose that you have a patient reported to you with a high fever. This is Celsius 40.5. You give a treatment, and after the treatment, it drops dramatically to all the way to here. But you could ask yourself, is my treatment sufficient? Because if you read out here, it's above 38, so it's not normal yet. And that is what I think is sort of ignored in people that like to do CBTI, like our team, we do it also a lot. Often we are not there yet, and we have to increase effectiveness. So can we do more than only CBTI? And this is something we tried. iSleep is an online treatment of insomnia, and we thought about, can we add things like addressing the biological clock, which tries to keep you asleep during the night and tries to keep you awake during the day? Can we help the biological clock by bright light, by scheduled exercise, or by scheduled warm baths? And we call that chronotherapy. And this is something that we tried, and this is what we found. So we had two groups. They both got cognitive behavior therapy for insomnia. And this is the pre-assessment, and the groups are not different, and that's because we randomized them. Then we put them into cognitive behavior therapy, and on the left, and then measure again their sleep efficiency. On the left, you see what happens with cognitive behavior therapy. On the right, you see what happens if we add chronotherapy to that intervention. Well, it's clear to see that it doesn't make it any better. So sleep efficiency doesn't increase significantly better if we add chronotherapy to cognitive behavior therapy. But then we did a follow-up, again 6 weeks later, and here we started to see differences. So in the people that had only cognitive behavior therapy, we saw that part of the benefits for sleep efficiency already dropped. But in the people where we also tried to boost their biological clock, they were better in maintaining the effects that were induced by cognitive behavior therapy for insomnia. And then we thought, okay, what will happen with depressive symptoms? I gave you the hypothesis that somehow insomnia maybe get in the way of resolving mood problems. And we just finished a 1-year follow-up study where we gave people treatment as usual, which is, well, treatment as usual is basically no treatment. We followed them up for a year, and we had high-risk insomniacs, so we did that in insomnia subtypes that I told you about. And we saw indeed that if anything, their depressive symptoms increased during the follow-up year. If we gave them cognitive behavior therapy for insomnia, that is the red trace here, we see that depressive symptoms decrease. Others have reported that also. If we give chronotherapy only, we see that it does nothing for depression. But if we give the combination, and again late, so we have to be patient to find these effects, again late, we find benefits of the combined treatment. And we did another follow-up study, even 3.5 years later, in the study that I showed you on the previous slide. And there we found that for the people that had no past intervention, this was 3.5 years after that other study, 9 out of 21 people had developed a diagnosis of depression or dysthymia. If people only had cognitive behavior therapy for insomnia, it was about the same. And then if they had a combined treatment, so CBTI either with light or with scheduled activity or with baths, they were much better in keeping their depression symptoms and depression diagnosis low. So this was my story. I think I told you a lot. Maybe review it if you can. And I think we have to move forward. We have a hypothesis now. As will turn out for most hypotheses, 30 years from now, we will know that it's not that simple as I sketched to you. But I think we have a hypothesis that we can work on together with clinicians and animal researchers, many people. So this is my team and some people that I collaborated with. And I'd like to thank you for your attention.